Welcome to the Deep Dive Lab, where we dive deep into fascinating science stories filled with surprising discoveries, unexpected twists, and thought-provoking ideas. So to start us off today, I want you to just imagine for a second that you've been hired to film a live theatrical play.
Okay, I'm with you.
Right, and it's this beautifully intricate performance, dozens of actors moving in perfect synchronization on the stage.
Sounds great.
Yeah, but there is one major problem. The theater is pitch black.
Oh.
Yeah, and your only piece of equipment, your only way to see them, is a spotlight that is so intensely powerful that the second you point it at an actor, it literally fries them on stage.
Oh, wow. Okay, that is a dark image.
It is, right.
Yeah.
But that dark, tragic scenario is basically the exact dilemma that scientists face every single day when they try to look inside living cells.
Yeah, it sounds like a strange thought experiment, but physically and mathematically, that is the exact barrier limiting modern cellular biology. I mean, we've developed these miraculous super-resolution fluorescence microscopes. They can see structures smaller than a single virus.
Which is incredible.
It is, but they require bombarding the sample with high-intensity lasers.
Right, which gives us this breathtaking image for, a fraction of a second, right before the laser just completely bleaches the chemical sample.
And it generates toxic free radicals that kill the cell.
So it's phototoxicity.
Phototoxicity, yes. We are destroying the very life we want to observe just by looking at it.
Which brings us to the core mystery we are unraveling today. We are looking at a groundbreaking new paper published in Nature by a team of researchers from Zhejiang University and Harbin Institute of Technology.
Right, Zhu, Zhang, Guy, and their colleagues.
Yeah, and the question they set out to answer is really profound. Like, how do we watch the intricate, nanoscale dance of life without destroying the dancers in the process?
And to solve that, this team realized they couldn't just, build a slightly gentler laser.
It wouldn't be enough.
No, they had to abandon external light altogether. They basically had to leave the theater in the dark and figure out a way to let the actors light themselves up.
Light themselves up, okay.
Yeah, and getting to that point required dismantling a massive assumption in optical physics.
Well, I really want to dig into that assumption because, biologists have known about the destructive nature of lasers for a long time now.
Absolutely.
Decades.
So it seems like the obvious pivot would be to use chemical luminescence. I mean, we see it in nature all the time, right?
Right, like the glow of a firefly.
Exactly. Or even just the chemical reaction you get when you crack a glow stick. It requires no external lasers, so it shouldn't cook the cells. So, okay, let's unpack this. If chemical luminescence is perfectly safe, and we've understood the basic chemistry of glow sticks for ages, what was stopping scientists from just, pouring this stuff onto a cell and looking through a microscope?
Well, it comes down to a physical barrier. It's known in the field as the low photon budget.
The low photon budget.
Okay.
Yeah, reaction-based luminescence is just unbelievably dim compared to the sheer volume of photons you get by bouncing a high-powered laser off a fluorescent molecule.
Okay.
So if you want to capture a single usable image of a cell using only chemical luminescence, the microscope needs to leave its camera shutter open for a painfully long time.
How long are we talking?
We are talking tens of seconds just to accumulate enough photons to render one single frame.
Tens of seconds for one frame.
Yeah.
Wow. I mean, that makes sense if you're photographing a rock, but a cell it's like a bustling microscopic city.
Exactly. Things are constantly moving.
So leaving a shutter open for 30 seconds on a moving target, you're just going to get a long exposure photograph of a busy street at night. You don't see the individual cars or the people walking.
No, you just get this blurry, glowing smear across the image, and that blur completely destroys your spatiotemporal resolution.
Because everything's moving.
Exactly. And because of that exact phenomenon, the scientific consensus for years was that achieving super resolution using chemical reactions was just physically impossible.
Because you were trapped behind that optical diffraction limit.
Right, the limit which dictates that you can't see distinct structures smaller than about 200 nanometers.
So lasers could break that 200 nanometer limit,
Yes.
but they killed the cell.
Yes.
And chemicals kept the cell alive. But the images were just way too blurry to break the limit.
It was the ultimate biological catch-22.
Wow. Okay, so how did this team actually break it? Because you can't exactly tell the internal organelles of a cell to just stop moving while you take a long exposure photo.
No, you can't stop the movement, but you can change how you process the light.
Okay, how so?
The breakthrough came when the researchers completely shifted their conceptual framework. They stopped looking at that chemical glow as a continuous, blurry smear.
Because it's not continuous.
Right. At a quantum and molecular level, reaction-based luminescence is actually a stochastic, highly dynamic process.
Stochastic, meaning random.
Yes, it is intrinsically driven by individual molecules reacting and emitting a photon, one by one.
Okay, so it's not a steady glow at all. It's more like a microscopic popping effect.
Yeah.
Like a sparkle.
Exactly, a very sporadic sparkle. And if you have a sophisticated enough computational framework, you can isolate those individual blinks in space and time before they blend into a blur.
Wow, okay.
So the team engineered this computational method. They called it
RIED.
Yeah, it stands for Reaction-Enabled Imaging via Entropy-Weighted Correlation combined with deconvolution.
Okay, that is a heavy wall of jargon. Reaction-enabled imaging via entropy-weighted correlation. Let me try to map that onto a physical image.
Go for it.
So if our living cell is a pitch black stadium, right?
Mm-hmm.
And the luminescent molecules are a crowd of thousands of people holding sparklers.
I like that.
Instead of taking one 30-second exposure and getting a giant lob of light, RED is taking ultra-fast, rapid-fire video frames. So most of the frame is pure blackness, because there are no lasers lighting up the stadium.
Right.
But for a tiny fraction of a second, one single sparkler goes off.
And because you have zero background light, the signal-to-noise ratio of that single spark is incredibly high. It is a brilliant point of contrast against absolute darkness.
Okay, but hold on. Cameras still have inherent static, right? Like sensor noise.
Oh, absolutely. Digital noise is a huge factor.
Right, so if you are taking rapid-fire video in the absolute dark, how does the algorithm know if it's looking at a real chemical sparkler or just a random digital artifact on the camera sensor?
Right, and that has to be where the entropy-weighted correlation comes in.
Okay, explain that to me. What is entropy doing here?
That is the genius of the RIED algorithm. So in information theory, entropy is essentially a measure of randomness or uncertainty.
Okay.
Random camera static has high entropy. It's completely chaotic and unpredictable.
Right, so it's just random speckles.
But a real chemical reaction emitting photons from a specific physical structure in the cell, that drops the local entropy.
Because it's coming from a real structured place.
Exactly. So the algorithm scans these rapid frames, basically mapping the spatial entropy to hunt for those localized drops.
That is so smart.
Yeah, it mathematically filters out the chaotic noise and isolates the true signals.
So it finds the true clusters of light, but a cluster of light is still, a cluster.
Yeah.
If we are trying to break the diffraction limit and see structures smaller than 200 nanometers, how do you pinpoint the exact molecule?
Well, that requires the next step, which is cross-correlation and deconvolution.
Okay.
Once the entropy filter finds the true sparks, the algorithm compares the sequential video frames.
To see how they change.
Yeah, by analyzing how the intensity of that spark fluctuates over time. It's blinking behavior, basically.
Oh, okay.
They can mathematically calculate the exact origin point of the photon emission.
Wow.
They aren't just taking a picture, they're computing a high-resolution map from sparse, blinking data.
That is a staggering piece of software engineering.
It really is.
But it does bring up a pretty practical biological problem. If you have this algorithm that can track sparks in the dark, how do you actually trigger the sparkler?
Right.
Like, you can't use a laser, so what is the ignition switch?
So to prove the algorithm worked, they first turned to something called electrochemiluminescence, or ECL.
ECL, okay.
Right. This involves triggering the chemical glow using an electrical potential.
Electricity.
Yeah. You place the cell near an electrode, apply a specific voltage, and it initiates the luminescent reaction.
Wait, hold on. You're talking about running an electrical current directly into a biological sample.
Yeah.
Doesn't shocking the cell fry it just as badly as hitting it with a high-powered laser?
Well, it would if the voltage were permeating the whole sample.
Right.
But ECL has a unique property called high surface sensitivity.
High surface sensitivity.
Yeah. The electrical trigger only initiates reactions within a few nanometers of the electrode's surface.
Oh, I see.
It doesn't penetrate deeply, so it doesn't cook the entire cell. It's an incredibly localized ignition switch.
That makes sense.
But there was a catch. Historically, ECL inside cells was still too dim to map out internal structures clearly, even with advanced algorithms like RE.
So the algorithm was hungry for more data, and the standard chemical reactions just weren't feeding it enough sparks.
Exactly, which meant the researchers had to hack the chemistry.
Hack the chemistry. Okay, here's where it gets really interesting. What did they do?
They took a commonly used co-reactant chemical called TPRA, and they swapped it out for a completely different molecular co-reactant called bistress.
Bistress. And the paper notes that by doing that, they boosted the brightness of the cell's glow by a massive amount, right?
Yes, an astonishing 1,000-fold increase in brightness.
Wait, really? 1,000 times?
1,000 times more light.
You can't just drop a new chemical into a cell and get 1,000 times more light without some heavy molecular physics going on. What is bistress actually doing differently?
It fundamentally comes down to the lifespan and stability of chemical radicals.
Radicals.
Right. So when the electrode triggers the reaction, it turns the co-reactant into a highly reactive radical.
Okay.
For the old chemical, TPRA, those radicals decay in mere microseconds. They are so volatile that they basically fizzle out before they can travel very far from the electrode.
Oh, so they die before they can even find a luminescent probe to interact with and create a spark.
Precisely.
Wow.
But the bistress radical has a much longer intrinsic lifetime.
So it sticks around longer.
Yeah. Because it remains stable longer, it has a vastly extended diffusion distance. It can physically travel much deeper into the cell's cytoplasm.
Navigating through all the internal cellular junk.
Exactly, navigating through the intracellular matrix. And that drastically increases the mathematical probability that it will collide with a luminescent tag and emit a photon.
Oh, wow. So that extended lifespan is what yields the 1,000-fold increase in signal.
That's it.
That's brilliant. So with 1,000 times more light and the RIED algorithm basically slicing through the blur, what did they actually see when they ran the experiment?
Well, they imaged microtubule filaments.
Those are like the tiny structural scaffolding inside the cells.
Exactly. And they imaged them at a resolution of 97 nanometers.
97 nanometers.
And they rendered it in 3 dimensions.
That is wild. They completely shattered that 200 nanometer physical diffraction limit.
Yeah.
And they did it relying solely on chemistry and math. No destructive lasers.
Yep. It was a monumental proof of concept.
Wow.
However, there's still an issue. ECL, because it relies on that electrode surface to trigger the reaction, is primarily used on fixed or dead cells.
Oh, right, because you have to pin it to an electrode.
Right. It gives you pristine structural maps, but it doesn't let you watch the living biological theater.
Ah, and that was the whole goal, to watch the dance without killing the dancers.
Exactly. So to achieve that, they had to take the RIED computational framework and pair it with bioluminescence, or BL.
Bioluminescence. Okay, so this is the firefly technology.
Yes, exactly.
So we are moving from dead cells pinned to an electrode to free-living, breathing cells. How do you ignite the sparklers without electricity?
They engineered a specialized bioluminescence resonance energy transfer system. It's known as a BRET system.
A BRED system. Okay, inside live cells.
Inside live cells.
Resonance energy transfer. Let's break that down for a second, because it sounds like we are talking about energy moving in waves, sort of like acoustic resonance between tuning forks.
You know, the tuning fork is actually a brilliant way to conceptualize it.
Really?
Yeah. So in a BRET system, you have an enzymatic chemical reaction that produces energy.
Right.
But instead of just releasing that energy as a dim standard photon, it relies on dipole coupling.
Okay.
If a specific fluorescent molecule is sitting incredibly close by, the energy transfers over to it without any radiation.
Oh, I see.
Yeah.
Just like striking one tuning fork causes a nearby one to vibrate?
Precisely. That second molecule acts as a highly efficient antenna broadcasting a much brighter, clearer light.
That's incredible. So the chemical reaction does the heavy lifting of generating the energy, and the antenna amplifies it, all organically inside the living cell.
Exactly.
And when they applied the RIED algorithm to this live cell setup, what happened?
They mapped out microfilaments, the endoplasmic reticulum, and mitochondria at around 117 nanometer resolution.
All while the cell is completely alive.
Completely alive and unharmed.
I want to ground this for a second, because we are throwing around incredibly small numbers here. 97 nanometers, 117 nanometers, and that absolutely proves they broke the optical limit.
It does.
But, you know, for the average person listening, why does this specific leap matter? Why do nanoscale photos of mitochondria impact the broader world of medicine and biology?
Well, the impact isn't just in the sharpness of the image, it is in the dimension of time. Yeah, it is an endurance test. We really have to look at what this technology allows biologists to do that was literally impossible yesterday.
Okay, give me an example.
Let's look back at one of the best live cell super resolution laser techniques currently available. It's called SIM, or structured illumination microscopy.
Right, the spotlight that fries the actors.
Exactly. When researchers used SIM to continuously observe live mitochondria, the high-intensity lasers immediately started generating reactive oxygen species.
Toxic stress.
Yes, the cell essentially begins suffocating on toxic stress. Within 18 minutes of continuous observation, the fluorescent proteins bleached out, the signal died, and the cell was fatally damaged.
18 minutes. Wow, I mean, you can't even get a pizza delivered in 18 minutes, let alone watch a cell undergo complex biological changes like tissue repair or an immune response.
It's a tragic short film.
It really is.
But now, look at RED-BL. Because it relies on gentle internal chemical resonance, there is zero phototoxicity.
Zero.
None. No reactive oxygen species are generated by an outside laser.
Because there is no outside laser.
Exactly. The researchers set up the system on live cells, and they just let the camera run. They continuously filmed the intricate dynamics of mitochondria in super resolution for 41 hours.
Wait, 41 hours.
41 hours.
The scale of that difference is staggering. Going from 18 minutes to 41 hours. I mean, that is the difference between watching a frantic 3-second movie trailer right before the theater projector catches on fire versus sitting down and binge watching a 4-season television series.
Right.
You actually get to see the entire character arcs develop and shift and resolve.
And because they had that 41-hour window, they witnessed a cellular phenomenon that is notoriously difficult to capture at the nanoscale over long periods.
Well, what is it?
They actually observed mitochondrial transfer.
Wait, mitochondrial transfer? Like organelles physically leaving one cell and migrating to another.
Yes.
But we always hear that classic textbook phrase, right? Mitochondria are the powerhouse or the battery packs of the cell. Are you saying cells are actively trading their battery packs? Why would a cell just give away its energy source?
Well, it is a critical, the somewhat enigmatic mechanism for intercellular communication and survival.
Really?
Yeah. Cells will literally build physical bridges, like tunneling nanotubes, to pass these organelles to neighboring cells.
Wow.
A healthy cell might transfer mitochondria to rescue a damaged neighbor that's trying to heal tissue.
Oh, that's amazing.
It is. Or, in a much darker scenario, cancer cells within a tumor microenvironment will actively hijack these bridges to steal mitochondria from surrounding healthy tissue.
To fuel their own growth.
Exactly, fueling their rapid growth.
That is wild. So we are basically watching microscopic highway robbery.
Yes, and with RIED, for the very first time, they could track individual mitochondria making this journey for hours on end.
And what do they find out by watching them?
By analyzing the data, they discovered that this transfer doesn't just happen one way, it occurs in two distinct modes of travel. And they classified these mathematically by looking at something called the diffusion exponent.
A diffusion exponent. Okay, I know that in physics, diffusion is usually just random movement, right? Like a drop of dye spreading out in a glass of water.
Mm-hmm.
What does the exponent actually tell us about how a mitochondrion is moving?
It tells us the level of intent behind the movement.
Intent.
Yeah. So a diffusion exponent of 1 represents pure random Brownian motion, like a drunk person stumbling aimlessly.
Okay.
Anything greater than 1 means the movement is directed, or super diffusive.
Ah, I see.
The first mode they found, which they called direct motion, had a diffusion exponent of 1.34.
So it's moving with a purpose. It's not stumbling. It's getting on the highway and driving straight to a destination.
Exactly. Biologically, this means the mitochondrion has likely latched onto the cell's cytoskeleton.
Like the internal structural highways.
Right, and it's being actively dragged forward by molecular motor proteins like kinesin. It's high-speed, uniform transit.
Okay, that makes sense. But you said there were 2 modes. What was the second one?
This second mode, indirect motion, was much more complex. It had a diffusion exponent of 0.96.
0.96. So it's less than 1. It's subdiffusive.
Yes.
Meaning it's moving slower than random chance.
It means it is getting constrained or trapped.
Oh.
The researchers watched these mitochondria move slowly, then stall completely for long periods before suddenly shifting back into directed motion toward the recipient cell.
It's taking the back roads, hitting stop signs, pulling over, getting stuck in traffic.
Exactly.
What is the biological traffic jam that causes a mitochondrion to just stall out like that.
Well, that is the beauty of this observation. It forces us to ask those exact questions.
Right.
The stalling could be the organelle getting physically wedged in a dense meshwork of actin proteins. Or perhaps it's pausing to switch from one molecular motor to another at a cytoskeletal intersection.
But the point is, RED allows us to actually see the traffic jam happening in real time at the nanometer scale without killing the cell while we watch it.
That is the crucial part, yes.
It fundamentally shifts what we are capable of discovering. I mean, let's pull all of these threads together because the sheer density of this breakthrough is massive.
It really is a lot to take in.
So what would you say are the ultimate takeaways from this deep dive?
I'd say the first one is that the optical diffraction limit is no longer an absolute barrier for biocompatible imaging.
Okay.
By treating chemical luminescence not as a continuous blur, but as a stochastic blinking process, we can completely bypass the need for destructive lasers.
So we don't have to fry the actors to see the play.
Exactly. Second, the RED algorithm is just a masterclass in computational imaging. By mathematically hunting for drops in spatial entropy, it can pull high-resolution structural maps out of what basically looks like pure, noisy darkness.
Yeah, that entropy filter is brilliant.
And third, pairing this software with advanced chemistry, like extending the lifespan of bistrous radicals or utilizing those Brett tuning forks.
Right.
It opens the door to ultra-long-term observation. We literally went from 18 minutes of toxic observation to 41 hours of harmless, continuous filming.
Which feels like one of those true paradigm-shifting moments in science.
Mm-hmm.
But as we've walked through this, something even more profound kind of stands out to me.
Oh, yeah?
Yeah, like traditional laser microscopy is brilliant, but it's ultimately just a map of physical positions. You shine a laser at a fluorescent tag, and it bounces back to say, hey, I am located here.
Right, it gives you coordinates.
Right.
Yeah.
But RED doesn't bounce light off an object. The light only exists if a specific chemical reaction is actively taking place.
Yes.
The molecule has to be working to emit a photon. We aren't just looking at the physical position of an organelle anymore. We are literally visualizing its chemical activity.
And that right there is the philosophical core of this paper. We are transitioning from tracking where biological components are to witnessing the chemical labor they are actually performing.
Wow.
If the chemistry stops, the light goes out.
Going back to that theater analogy we started with, we haven't just figured out a way to light up the stage without burning the actors.
Right.
We have engineered a stage where the actors only emit light because they are actively performing their unique roles.
Beautifully said.
It is an entirely new way of witnessing biology.
Mm-hmm.
And it leaves us with an incredibly provocative reality to confront.
What's that?
If we can now watch the actual chemical conversations of living cells undisturbed for 41 hours, what happens when we turn this technology on a tumor? We might finally be able to watch in real time and in the dark exactly how a cancer cell chemically deceives a healthy immune cell into ignoring it.
The microscopic theater has finally been left in the dark, and the cells are ready to speak. We just have to watch.
A huge thank you to everyone listening for joining us on this deep dive. If you want to keep exploring the absolute cutting edge of what's possible, make sure to follow and subscribe. We'll catch you next time.